that introduction, and thank you, Dan and Andrew, for the opportunity to speak today. So I'm going to talk to you today about how we might move away from the traditional approach to drug discovery using animal testing and towards an animal-free future. Sorry. Project a bit more. So I'll start with some background on why animal testing was introduced, then show you how the pharmaceutical industry has fared over the last 20 or so years from the current drug discovery paradigm, where the problems might be, and how we can solve them by changing the approach and by adopting new technologies. So the basic principle behind the introduction of animal testing was to protect human life. 1938 saw the introduction of the first formal requirement in law to prove the safety of a new compound following the death of 100 people from kidney failure after ingesting this elixir sulfonamide, which contained toxic diethylene glycol. The requirement for animal testing was strengthened after the thalidomide tragedy in the 60s and 70s, and new laws were introduced mandating that new chemical entities be tested on pregnant animals as well. The irony of this is that thalidomide is non-toxic to most animal species, so even if it had been tested in this way, the effects would not have been detected and it probably still would have been cleared for human use. So animal testing is required by law to protect human lives, but since its introduction there have been concerns raised about its ability to accurately predict human toxicity. A brief introduction to the drug discovery process then, for those who haven't come across it before. So here's a timeline for drug development, and it begins at year 0 with the filing of the patents and the screening of a library of about 10,000 compounds for effectiveness against a target and for cell toxicity. Those compounds which do the job and don't cause toxicity are passed on to preclinical screening in animals, which takes us up to about year 4 or 5, and successful drug candidates are then passed on to trials in healthy humans in the clinic and then into patients. But I just wanted to give you an idea of the timescale the drug discovery process works to. There's 10 to 13 years of R&D before a drug even gets to market, if it gets there at all. So, current opinion, an opinion shared by the new directive, is that the use of live animals continues to be necessary to protect human and animal health and the environment. However, of the new chemical entities reaching clinical trials, so they've gone through testing on cells in well plates, they've passed animal screening, 94% of drugs in that preclinical testing stage will either not be effective or will be found to cause human toxicity. On top of this, even those drugs which make it to registration can cause toxicity undetected during the drug development process, to such an extent that in 2011, 197,000 deaths due to adverse drug reactions were reported in the EU. The picture isn't any better for the pharmaceutical industry themselves. In fact, the industry is suffering an economic crisis. You've probably heard about the huge numbers of redundancies made in recent years by Pfizer, by GSK, by Unilever, with many more set to go in AstraZeneca's Alderley Park as they prepare to move to Cambridge. And here's why. Investment in drug R&D has increased over the years since 1990, and returns have diminished. Old patents are running out, and there aren't any new ones, or many new ones, to take their place. So from all this, we can safely conclude that the current testing strategy is inadequate and doesn't accurately predict the effectiveness or safety of new drugs. But why is that? Well, preclinical testing is not representative. Cell screening is done on small populations of cells, in isolation, in 2D culture, under static conditions, often with cell lines which are different to primary cells, so cells taken from the body, and this is not what cells experience in their natural environment. Animals are not humans. They metabolize drugs differently, they respond differently, so they can't provide a reliable indicator of what the response will be in humans. For example, when using animal models to develop cancer treatments. We have cured cancer in mice 7 times over, but the challenge still remains in humans. This was taken from a presentation by Bill Greenhalgh at our conference in May this year. Very interesting talk. So there's also the element of patients themselves, which adds an extra layer of complication. Responses to a drug can vary depending on genetic makeup, whether they have existing conditions, and interactions between new drugs and any other medication that they would take. So there are a variety of problems with the current approach, but all is not lost. It just means that we need to consider new approaches, for example those based on computing methods and alternative testing strategies. The requirement for animal models comes from inadequacies in vitro testing. Animals provide the ability to test drugs on more complex systems, systemic and whole organ systems, but there are now alternative technologies that make it possible to do this with human cells in a dish, so to speak, and get more accurate results than with animal testing. Also this idea of microdosing, where subtherapeutic levels of drug are administered to humans and a response can be safely measured. So what might the new approach look like? If you keep in mind the drug development overview I showed you earlier but flipped on its side, so we have time running in this direction. A new approach might look something like this. So utilising the huge amount of computing power we have available now to convert the wealth of information we have on signalling pathways, disease phenotypes, different genotypes, and drug target interactions into predictions of effectiveness and toxicity, so that before we even get to applying the drug on live cells, the library has been trimmed down and made far more selective, and the outputs are more likely to be successful down the line. Then we move into a system of better in vitro testing, using alternative cell culture models for systems, for organs, or for disease that will be able to give us a much more accurate picture of what reaction to expect in the clinic, which can then be tested by microdosing before moving to therapeutic doses in patients. And at the end of this process, we have a far better functional knowledge of how a drug works, enabling improved safety and efficacy and development of novel drugs. So I keep talking about alternative technologies, but what are these and what do they look like? So the aim of alternative technologies is to replicate the in vivo, the environment in the body in vitro, so that cells behave in the same way they do in the body when challenged with a drug. 3D scaffolds to mimic the architecture of our organs. Perfusion systems to model the flow of blood or lymph. Culture of different cell types together because no cell type in the body exists in isolation. This idea of primary or primary-like human cells to give the most relevant representation of activity and function, and a combination of all these elements to provide a complete picture of in vivo metabolism. So these technologies can be used to accurately model organ systems. I've included some data in this presentation so people can go over it at greater length when these slides are made available, but I'm going to go over these briefly now. And what I want you to take from them is the capacity for existing technologies, so not in development, these are commercially available technologies now, the capacity of these technologies to accurately replicate complex systems such as glucose and fatty acid homeostasis in the bloodstream controlled by the liver, and the function of the liver itself. So here we can see the improvement in cell division under nutrient perfusion in a 3D scaffold. This is a cross-section of a 3D scaffold, so under flow compared to in standard static conditions without perfusion. The improved metabolic function of cells enabled by this technology translates into more accurate prediction of drug toxicity. For diclofenac, which is an anti-inflammatory medicine, standard static culture predicted liver toxicity at a concentration 59 times higher than concentrations which cause toxicity in the clinic. And testing on rats predicted concentrations up to 39 times higher. So patients were given doses of diclofenac at levels predicted to be safe, which were in fact toxic. Cells in our quasi-vivo system detected toxicity at levels much closer to those in the clinic and with 12 times more sensitivity than standard in vitro methods. On top of the scientific arguments for change, there is also an economic argument. Here we have a breakdown of the current drug discovery process into percentage of funding allocated, and I've overlaid the figures in millions based on this conservative average of $1.5 million cost per drug developed. The majority of funding goes into clinical trials, this section here, almost 60% in fact, but nearly 25% is spent on preclinical testing, which is the part that involves animal testing. So, if we know that 149 new drugs were registered in the period between 2007 and 2011, that is, drugs which made it successfully all the way through. So, drugs which made it successfully all the way through this process and were approved for human use. If we apply this 94% failure rate, we can estimate that during the same time period, over 2,000 entities, new drugs, failed after being cleared by animal tests. So this equates to an estimated loss to the pharmaceutical industry of over $868 million. And that's with the conservative estimate, remember. I've seen figures bandied about that it could be as much as $12 million to develop a single drug, with an average of about 6 at the moment. So, following conversations with pharmaceutical contacts, we predict that by switching to more representative alternative models, something in the order of a quarter of these costs could be saved, increasing as models are improved and refined. So the benefits of moving away from animal testing and towards testing using alternative technologies could be huge. But how do we go about changing the existing drug discovery process? Realistically, this will require a great deal of investment. Funding bodies like NC3R's National Centre for Reduction, Replacement and Refinement offer grants for projects aiming to reduce or replace animal use, or refine procedures to reduce suffering or increase relevance. And their budget has been increasing year on year, but is still a fraction of the allocations given to other funding bodies such as TSB or BBSRC. What we really want to see is an increased investment from the industry that stands to gain the most from these new technologies, so the pharmaceuticals and CROs. There also needs to be more legislative support from governments. So going back to what Judy was saying earlier about responsiveness to what's happening in the scientific community, less reliance on lobby groups to get the message across. The new EU directive seeks to facilitate and promote the advancement of alternative approaches, but doesn't make any mention of how these provisions should be made. Engagement and communication are essential aspects to implementing any solution, and that's no different here. We as technology providers must engage with our users and obtain independent validation of our alternative approaches through bodies like ECFAM, the European Centre for Validation of Alternative Methods. So, the main barriers to the implementation of alternatives are the uncertainty and perceived risk involved in changing existing methodologies, which technology providers like ourselves can address with evidence of efficacy through conversations about the requirements of the pharma industry, so they're getting exactly what they need. A lack of development funding, without which it is difficult or impossible for SMEs to generate data on the effectiveness of their new technology. The scale of the investment required to validate these technologies is a significant barrier. So, pharmaceuticals are looking for validated solutions, but 10 years, €5 million is an unreasonable investment that needs to be reduced. The pharmaceutical industry must also embrace change rather than be suspicious of it, and be willing to work with technology providers to develop and validate alternative solutions. The current paradigm does not work. The drug discovery process is stagnating. Now is the time to reverse that. We have the solutions, we've got the technologies, now we need to work together to implement them. So what we have here is an opportunity to replace a broken system that is only going to get worse with a system that is not only cheaper, not only more ethical, not only more efficacious, not only safer, but also more scientifically valid. It will require investment and perhaps legislative encouragement, but this short-term investment will reap huge long-term gains. If we don't act now, revisit the requirement for animal testing, and address the application of these alternative models, we risk losing both the testing and the development of the alternative technologies to emerging market economies. So companies like GSK are already starting to move their testing into China, into India. So I'll just leave you with that thought.